Imagine a blueprint, right, for like a massive state-of-the-art skyscraper.
Okay, I'm picturing it.
We are talking about 3 billion lines of complex architectural code. And if someone handed you that blueprint and told you that buried somewhere on page 10,000, there was a single typo, just one letter in the wrong place, what would you think?
I mean, you probably assume it's no big deal, like maybe a light switch ends up wired on the wrong side of a hallway or something.
Exactly, because a structure that massive is designed to be robust. Statistically, one typo really shouldn't compromise a whole skyscraper.
Right, the building will stand perfectly fine.
But when you step into the realm of human biology, and specifically the genetics of how we age, that exact assumption completely shatters.
It absolutely falls apart because in human biology, depending on exactly where that typo is located, it doesn't just, misplace a biological light switch.
Right.
It actually has the power to fundamentally warp the structural integrity of every single cell in the entire body.
Which brings us right into our mission for today's deep dive. We are unpacking a truly incredible stack of sources for you today.
Yeah, we've got medical journals, clinical trial summaries, and the latest research straight from the Progeria Research Foundation.
And we are taking a deep dive into Hutchinson-Gilford progeria syndrome.
Often known simply as Progeria.
Exactly. But we aren't just looking at an ultra-rare pediatric rapid aging disease today. We are essentially taking a masterclass in how one single genetic typo rewrites human biology.
And maybe even more incredibly, how science is fighting back.
Yes, we are looking at cutting-edge therapies being developed right now that, well, they might eventually teach us the secrets of our very own aging process.
It's really a perfect example of how studying the rarest of conditions can unlock universal biological truths for everyone.
So true.
So we're going to trace this disease from its epidemiological roots, map out the exact molecular machinery that breaks down, and look at how researchers have built this rapidly evolving landscape of treatments.
Everything from repurposing old cancer drugs all the way up to revolutionary CRISPR base editing.
Yeah.
Okay, let's unpack this. Because to really understand how to treat progeria, we have to start with who it affects. And that means wrapping our heads around just how rare this condition actually is.
It is exceptionally rare.
Right, looking at the clinical data here, we are talking about an estimated prevalence of 1 in 20 million. There are only about 400 children worldwide living with this condition right now.
And the clinical progression for those children is, it's heartbreakingly rapid.
Yeah.
When babies with progeria are born, they appear perfectly healthy. There are no immediate red flags at all.
Which is so wild to think about.
It is. But usually within that first year or two of life, severe symptoms begin to manifest. They experience profound growth failure, a near total loss of body fat, hair loss.
Joint contractures.
Yes, joint contractures and severe osteoporosis.
And without treatment, the life expectancy is tragically short. The average is just 14.5 years.
Yeah, 14 and a half. And the primary cause of mortality is what we typically associate with elderly patients.
Fatal cardiovascular issues.
Exactly. We are talking about severe atherosclerosis, the rigid hardening of the arteries, which leads to heart attacks and strokes in teenagers and sometimes even young children.
It's just devastating.
Yeah.
And it's so hard to reconcile that massive systemic breakdown with the root cause. You mentioned that out of the 3 billion letters in the human genome, this all traces back to one wrong letter.
Just one.
A single C swapped out for a T.
Specifically, it's a point mutation in the LMNA gene pronounced lamin A, which is located on chromosome 1q22. And there's a crucial term in the literature you'll see here. It's a de novo mutation.
De novo, meaning it's new, right? So the parents aren't carrying this mutation and passing it down.
Exactly right. It is almost never inherited. It's a spontaneous transcription error that just happens during early cellular development.
Wow.
Out of nowhere, the exact mutation designated as C.1824C2T occurs.
It's like a genetic lightning strike. It's completely random. But how does that one strike, one tiny typo in an instruction manual, cause the entire cellular factory to break down? What is this LMNA gene actually supposed to be doing?
To answer that, we have to look at the structural mechanics of our cells. The LMNA gene is the blueprint for proteins called lamin A and lamin C.
Okay.
These proteins are the critical structural components of the nuclear lamina. If you think of a cell's nucleus, the vault that holds all our DNA.
Right, the central vault.
The nuclear lamina is a dense meshwork, a biological scaffolding that sits just inside the nuclear membrane. It holds the vault together, gives it its shape, and protects the DNA inside.
Okay, so lamin A is basically the load-bearing wall of the nucleus.
That's a great way to put it. And the sources actually highlight how sensitive this specific genetic neighborhood is.
How so?
Well, while that 1C to T typo causes classic progeria, there are non-classic forms caused by other pathogenic variants in exon 11 or intron 11 of that same gene.
Oh, interesting.
There is even a related condition called restrictive dermopathy, or RD, that's a highly lethal neonatal condition caused by either a homozygous mutation in the LMNA gene or a mutation in the gene that produces the cellular tools that work on lamin A.
So if you mess with the scaffolding blueprint in literally any way, the consequences are catastrophic.
Exactly.
But let's stick to the classic HGPS. We have the typo. We know it affects the load-bearing walls. But how exactly does it go from a bad blueprint to a broken cell?
This is where we need to trace the molecular pathway. The sources provide a really detailed infographic on this, and breaking it down is essential to understanding the cure.
Let's do it.
In a healthy cell, the LMNA gene doesn't just instantly spit out a finished protein. It produces a raw precursor called pre-lamin A.
Like a rough draft.
Right. And for pre-lamin A to become mature functional lamin A, it has to undergo a highly specific 4-step modification process.
Okay, so the factory has built the raw part, but now it needs to be customized before it can be installed. What's step 1?
Step 1 is called farnesylation. The cell uses an enzyme to attach a lipid anchor, a farnesyl group, to the very end of the protein.
A lipid anchor.
Yeah, and this anchor is highly hydrophobic, meaning it violently repels water. Because the inside of the cell is watery, this anchor naturally drives the protein to embed itself firmly into the fatty membrane of the nucleus.
Oh, that makes total sense. It's like attaching a heavy magnet to the protein so it gets pulled right to the wall where it needs to be installed.
Exactly with step 2.
Step 2 involves a specific enzyme called ZMPSTE24.
Catchy name.
Right. Just think of it as molecular scissors. It snips off a tiny piece of the protein right next to that anchor.
Okay, snip. And step 3.
Step 3 is carboxymethylation, performed by an enzyme called ICMT. This basically caps the end of the protein to chemically stabilize it.
Got it. Anchor attached, a little trim from the scissors, and a chemical cap. What's the final step? Step 4 is the absolute most crucial part. Once the protein is safely embedded in the nuclear membrane and correctly positioned, those exact same ZMPSTE24 scissors come back and make a final, deeper cut.
Wait, what are they cutting now?
They snip off the entire farnesyl lipid anchor.
Hold on, let me make sure I'm tracking this. The cell intentionally goes through all the trouble of attaching a lipid anchor, just to send the scissors back to chop it off.
Yes.
Why bother adding it in the first place?
Because mature lamin A needs to be free to move around and do its job dynamically within the scaffolding. It needs to be flexible.
Ah, I see.
If the anchor stays attached, the protein is permanently tethered to the membrane. It becomes completely rigid.
Ah, okay. It's like putting a heavy plastic shipping wrap around a piece of furniture to transport it. You need the wrap to get it into the house, but once it's in the living room, you have to cut the wrap off, or you can't actually use the furniture.
What's fascinating here is that your analogy perfectly highlights the mechanical failure in progeria. Yeah. So let's look at what that single C to T typo actually does. It activates what geneticists call a cryptic donor splice site.
A cryptic splice site. Okay, let's translate that. RNA splicing is basically like a film editor, right?
Mm-hmm.
The editor's job is to cut out the boring, non-coding parts of the genetic movie and splice the important scenes together.
Exactly. And that typo creates a fake cut here sign. It tricks the film editor.
Oh, no.
Because of that one wrong letter, the cell's machinery accidentally snips out 50 crucial amino acids from the final protein instructions.
So 50 frames of the movie just vanish.
And here's the devastating part. That missing chunk contains the exact recognition site for the final scissor cut in step 4.
Oh, wow. So the cell attaches the sticky lipid anchor to move the protein to the wall, but because of the typo, the scissors literally can't find the spot to cut the anchor off.
Precisely. Because that final cut never happens, the cell produces a mutant, permanently anchored protein called progerin.
Progeria.
Yes, and progerin is incredibly sticky, rigid, and highly toxic. It accumulates inside the nuclear membrane, completely unable to detach.
And if you have all this rigid, sticky protein building up on the inside of the nucleus, what does that actually look like for the cell?
It causes massive structural failure. Under a microscope, you can literally see the architecture of the nucleus warp. It becomes lobulated or severely misshapen, like a crumpled ball.
That sounds awful.
It is. This structural failure triggers a massive cascade of downstream damage, chronic DNA damage, accelerated shortening of telomeres, mitochondrial dysfunction, and premature cellular senescence.
Meaning the cells permanently stop dividing and basically age out prematurely, which
Exactly.
perfectly explains why teenagers are having heart attacks. The blood vessels are lined with cells that have to endure massive mechanical stress from pumping blood. If their internal scaffolding is rigid and crumpled,
They can't handle the stress.
right, those cells die fast, the arteries harden, and the whole cardiovascular system breaks down.
The load-bearing walls fail, and the system collapses.
Okay, so if we know this exact four-step process, and we know exactly where it breaks at step 4, because the scissors can't make the final cut, how do we fix it?
Well, mapping that pathway is what gave scientists their first highly specific targets. If progerin is toxic because it's permanently farnesylated, because that lipid anchor makes it sticky, researchers realized they could intervene by blocking those modification steps earlier in the chain.
Oh, so they didn't have to wait for step 4.
Right.
So if you can't cut the anchor off, maybe just prevent the anchor from ever being attached in step 1.
Exactly. This led to the drug repurposing era from 2003 to 2010. Scientists looked at cancer drugs called farnesyl transferase inhibitors, or FTIs.
Cancer drug.
Yeah, these were originally developed to stop cancer-causing proteins from getting lipid anchors. They hypothesized they could use them to stop progerin from getting its anchor.
And they actually work.
It did. This research culminated in 2020 with the historic FDA approval of lonafarnib, sold under the brand name Zokinvy.
Wow.
It became the very first and currently only FDA-approved drug for HGPS.
That is a massive milestone. I mean, going from discovering the gene in 2003 to an FDA-approved treatment is incredibly fast in the medical world.
Yeah.
How much does lonafarnib actually help these kids?
The clinical trials showed that lonafarnib extended average survival by about 2.5 years.
Okay, that's significant.
And follow-up data has suggested it could extend lifespan by up to 4.3 years for some cohorts over an 11-year period. It dramatically improves weight gain, bone density, and arterial function.
But reading through the clinical data, there's a significant catch, isn't there?
There is. While it extends life, it doesn't fully halt the underlying disease progression. The cell is still producing progerin, it's just missing its anchor.
Right.
Furthermore, because you are blocking a fundamental cellular process, the drug comes with severe side effects. Up to 80% of patients experience significant gastrointestinal issues like chronic nausea and diarrhea.
Oof. So it's an incredible lifeline, but it's not a comfortable cure. Which makes sense. If blocking the anchor in step 1 causes severe nausea, did researchers try attacking the protein from other angles?
Absolutely. The sources outline a whole landscape of other small molecule inhibitors. For instance, there are ICMT inhibitors.
What do those do?
These block step 3, the carboxymethylation capping step. In mouse models, preventing that cap successfully makes a progerin less sticky, delocalizing it from the nuclear membrane and extending lifespan without the same level of toxicity.
So if you can't comfortably stop the anchor, stop the cap. What else?
Then there's a completely different strategy, mTOR inhibitors like everolimus and rapamycin. These don't try to stop progerin from forming at all. Instead, they activate a process called autophagy.
Autophagy. Okay, that's the cell's internal garbage disposal system, right?
Exactly. The idea is to upregulate the cell's natural recycling machinery to rapidly clear out the toxic progerin before it can warp the nucleus.
That's pretty clever.
There are actually phase I and II clinical trials happening right now that combine everolimus with lonafarnib. The goal is to attack the mutant protein from both ends, stopping it from getting an anchor, while simultaneously accelerating its disposal.
I also see something in the notes here about lumino binders.
Yes, compounds like JH4 and a much newer orally administered drug called progerinin. Instead of messing with the cell's modification enzymes, these small molecules act like a biochemical straitjacket.
A straitjacket.
Yeah, they bind directly to progerin itself, neutralizing it so it can't interact with and damage the nuclear lamina. Progerin is currently in phase I trials, and in mice, it showed roughly a 50% lifespan extension,
Wow.
which actually demonstrated better results than lonafarnib alone.
So what does this all mean for you listening? We have some incredibly clever strategies here. We are essentially throwing sand into the gears of the protein factory, or boosting the cellular garbage disposal, or putting a straight jacket on the toxic progerin.
Right.
But if I'm understanding this right, all of these therapies are just managing the chemical fallout. We still aren't fixing the original typo in the blueprint.
This raises an important question, and you've hit on the exact limitation of small molecules. As brilliant and life-saving as they are treating the symptoms at the protein level. To truly cure the disease, you have to go upstream.
You have to correct the instructions themselves.
Exactly.
And that takes us to the next frontier, targeting the RNA and the DNA. How is modern science moving from managing the fallout to actually fixing the blueprint?
The first step upstream is at the RNA level. Remember our film editor analogy?
Yeah, the editor cutting the frames.
Researchers are using antisense oligonucleotides, or ASO therapy, to intervene during that editing process. A drug called SRP-2001 binds specifically to the mutated sequence in the messenger RNA and physically blocks that fake cut here sign.
Oh, that's brilliant. It acts like a piece of tape over the typo, forcing the cell's editing machinery to ignore it and splice the instructions correctly.
Exactly. And in mouse models, this ASO therapy increased lifespan by 50 to 60%.
That's huge.
But the ultimate frontier, the true holy grail of genetic medicine that the Progeria Research Foundation is pursuing, is fixing the DNA itself.
I've been waiting for CRISPR to come up.
And the progress here is nothing short of astonishing. But they aren't using traditional CRISPR. Traditional CRISPR acts like molecular scissors that cut the DNA double helix, which can be risky and cause unwanted mutations.
Right, because you're literally slicing DNA.
Instead, researchers are using a breakthrough technology called an adenine base editor, or ABE.
How does that differ from the molecular scissors?
Think of a base editor less like scissors and more like a highly precise pencil eraser. Researchers were able to go into the genome and chemically convert that mutated AT base pair back into a normal GC pair.
Wait, they just changed the letters?
Yeah, they didn't cut the DNA, they just rearranged the atoms to rewrite the single typo.
In living animal?
Yes. A single injection of this base editor in Progeria mouse models resulted in a massive 2.5 times increase in lifespan.
Are you serious?
The mice went from living 215 days to 510 days, which corresponds to the start of old age in healthy mice.
A 2.5 times increase from a single injection. That is staggering.
And crucially, it completely preserved healthy vascular tissue, which, as we discussed, is the main predictor of mortality in human patients.
Wow.
The Progeria Research Foundation is currently advancing this CRISPR-based strategy known as SAMPRO-2 toward human clinical trials. It represents a massive paradigm shift. We are moving from a chronic, daily drug that causes severe nausea to a potential one-time permanent cure.
Here's where it gets really interesting, though, because
Yeah.
among all this high-tech CRISPR and genetic base editing, there is a stunning biological secret hiding in plain sight in our sources.
There really is.
It turns out human brains are remarkably unaffected by progeria.
Yes, it's a fascinating medical paradox. The cardiovascular system is ravaged, the bones suffer severe osteoporosis, the skin ages rapidly.
But neurological function remains completely intact. The brain simply doesn't age prematurely.
And the reason why is just wild. The sources explain that the human brain naturally possesses a highly specific microRNA called miR9.
miR9.
And this miR9 naturally binds to the transcripts for laminin and progerin and totally silences them.
It's incredible. It prevents the protein from ever being translated and produced in those specific neurological cells.
Exactly. And we know this because the highest abundance of active miR9 is found in the cerebral cortex and the cerebellum.
So the human brain basically evolved its own highly specific spam filter that perfectly deletes this toxic message before it can even be read by the factory.
That's exactly what it is.
It's nature's own proof of concept for gene therapy.
If we connect this to the bigger picture, it validates everything researchers are trying to do therapeutically. The brain shows us that if you can silence or prevent the expression of progerin, you can completely protect the tissue from the disease.
Which is incredibly hopeful.
It proves that whether we use CRISPR-based editors, RNA blockers, or take inspiration from nature's own microRNA, a definitive cure is no longer science fiction. It is a biological reality that we are rapidly approaching.
It is so rare that we get to see a scientific story move this fast. I mean, in just over 20 years, we went from discovering the LMNA gene typo in 2003 to repurposing cancer drugs, to the historic FDA approval of lonafarnib in 2020.
And now we are standing on the absolute brink of permanent CRISPR-based editing cures.
It is a profound testament to the power of targeted research and the relentless push of organizations like the Progeria Research Foundation. But as we promised at the start of this deep dive, The implications of this work go far beyond the 400 children currently living with HGPS.
Right, it affects all of us.
Why should you, the listener, care about the mechanics of this ultra-rare disease? Because the sources reveal a mind-bending fact. Even in the cells of completely healthy, normal, elderly people who do not have that 1 in 3 billion genetic typo, this exact same cryptic splice site we've been talking about spontaneously activates over time.
That's right. As we age, our normal, healthy cells start to make occasional mistakes in splicing the LMNA gene.
The film editor gets tired.
Exactly. The editor gets tired and occasionally makes that exact same accidental cut. So healthy individuals start producing small amounts of progerin.
Crazy.
It builds up slowly over decades rather than months, contributing directly to the natural cellular senescence and cardiovascular stiffening we all experience as we get older.
So what does this all mean? If normal human aging involves the exact same toxic progerin buildup found in Progeria, it really forces you to ask something huge.
What's that?
Could the groundbreaking therapies being developed right now to save these 400 children, the CRISPR base editors, the RNA blockers, the cellular garbage disposal boosters, could they eventually become the key to how humanity slows down the biological clock for everyone?
It's a profound thought. By trying to save these children, the fundamental molecular machinery of human aging is being laid bare, step by step.
We started this deep dive talking about a skyscraper with a single typo in its blueprint. It turns out that one typo isn't just a rare anomaly. It's the key to understanding how the entire building eventually wears down over time.
And maybe, just maybe, it's teaching us how we can reinforce the foundation for good.
Something to ponder next time you think about the passage of time.
Definitely.
Absolutely. Thanks for joining us on this deep dive.